Hi, my name is Joe Wayman. I'm a researcher in the Meraldi lab at Cincinnati Children's Hospital. I'll be discussing my work in building gene regulatory networks from single-cell genomics data. So I'll first give an overview of the methods we use to infer gene regulatory networks, and then I'll show an application of these methods to identify important regulators in aging CD4 T cells. So this work is in collaboration with the Hildeman and Cunier labs at Children's. So to infer gene regulatory networks, we use a method called the Inferelator. So this method infers connections between transcription factors and the genes they regulate. So the inputs to this workflow are gene expression data, and some sort of prior information about known or estimated transcription factor gene interactions. So we often infer a prior network from chromatin accessibility data. So the Inferelator models gene expression as a linear function of transcription factor activities. So as a robust estimate of TF activity, rather than use TF transcript level, we use the expression of all of a TF's targets from our prior network. And we also apply an adaptive lasso that more heavily weights interactions present in our prior. It's able to identify interactions in our prior that are supported by patterns in gene expression. Is also able to learn new interactions strongly supported only by gene expression. So to generate a prior gene regulatory network, we typically use a combination of chromatin accessibility and TF motif scanning. So we will identify potential regulators of a target gene by scanning for TF motifs in open promoters as well as distal enhancers. If we have single-cell ATAC-seq, we infer these distal enhancers using a tool called Cicero that finds pairs of regions with correlated accessibility across cells, penalizing the distance between these regions. So we've applied this method of GRN Inference to identify important regulators in an age-associated immunosuppressive population of CD4 memory T cells, recently studied by our collaborators. So they've discovered this population of CD4 cells producing IL-10 with a T follicular helper-like phenotype that accumulate with age. So Tfh cells typically interact with B cells in germinal centers, and they play an important role in their selection and differentiation into plasma cells. And in this study, they show that these Tfh-like cells accumulate. We call them Tfh10 cells due to their production of IL-10. Their emergence is likely a counterbalance to chronically elevated inflammatory factors such as IL-6 in old age. So they also show that these Tfh10 cells accumulate after immunization in old mice, and blocking IL-10 signaling increased B cell responses in old mice. So to further study these Tfh10 cells and characterize their regulatory networks, we've generated single-cell RNA-seq and single-cell ATAC-seq using IL-10 GFP reporter mice. We've enriched for IL-10 positive cells. So we end up with about 40,000 cells in our data set, and after SROT integration and clustering, we've annotated our cell types based on markers in comparison with other single-cell data sets in aging T cells. And we've identified what we think are 3 clusters of Tfh10 cells. On the right, I'm showing how these cell populations change with age. And amongst the IL-10 positive cells, these Tfh10 cells populations all increase in old mice, along with a population of cytotoxic CD4 T cells. So we've combined our Inferelator workflow with a multitask learning approach to generate gene regulatory networks in young and old mice. So this method learns two networks simultaneously, one for young and one for old, by identifying interactions that are similar across age, as well as those that are unique to each age group. So this method gains statistical power by combining data sets and is used when trying to infer networks for systems expected to have some overlap in their gene regulatory networks. So we infer networks that describe regulation of all cell populations in our data sets. And then we can identify regulators that are important to each cell type, which I'll describe on the next slide. So here I'm showing you portions of the networks that are prominent in young TFH and old Tfh10 cells. So the squares represent TFs and circles are target genes. The colors represent relative gene expression. And then TFs can either activate or repress gene expression in our networks. So these subnetworks shown here show the regulation that makes old Tfh10 cells differ from normal TFH, young TFH cells. So Tfh10 have an absence of classic TFH regulator TCF7 that drives CXCR5 and BCL6 expression. And we have also identified regulators that drive these important cytokines IL-10 and IL-21. So we identify core transcription factors for each population. So these are TFs with enrichment of highly expressed cell type specific genes amongst their inferred network targets. So basically we find markers for each population, and then core TFs are those whose targets in our network are enriched for those genes. So if we zoom in, I can give a kind of clearer example. We find that network targets of FOXP3, an important regulator of Treg function, is enriched in genes highly expressed in Treg, as we would expect. And on the right here is we've found these cores for young and old. And on the right is the difference in core TF enrichments, where purple would define TFs that play a more important role in old cells and green more important in young cells. So we've identified here a group of TFs that are relatively more important in Tfh10 function in old cells, as well as a few that are more important in young cells. So we've built networks that describe regulation within these cells. Another area we're exploring is inference of networks describing interactions between cell types. So to do this, we've been using this recent tool called CellChat that infers communication between cell populations based on expression of pairs of receptor and cytokine signaling molecules. So, and we've integrated our data set of young and old CD4 T cells with age-matched cells from the spleen from this tabula muris cell atlas, and we've inferred possible interactions between cell types amongst young cells and amongst old cells. So these are the estimated outgoing and incoming signals for each cell type split by age across the top, and each cell-cell signaling pathway along the side. So we see that in old Tfh10 cells, similar to old Treg cells. These are interacting with macrophage and red blood cell progenitors through immunosuppressive cytokine IL-10. And then we've also identified some other cell-cell communications that are prominent in old Tfh10, such as CD6 ALCAM cell-cell contacts. We also see interactions that are common to many cell populations amongst old cells, including TNF light signaling. So we're still trying to understand these many cell-cell interactions that could be playing an important role in old Tfh10 function. So to summarize, using network inference methods from single-cell genomics data, we've identified important regulators of this immunosuppressive Tfh10 population. And going forward, our goal is to experimentally perturb intracellular regulators or intercellular signaling pathways that are important to this immunosuppressive function of Tfh10. With that, I'd like to thank my lab as well as our collaborators. And thank you for your attention.